So thank you, Melanie, and good afternoon or good morning, everyone. I'm pleased to introduce this wide discussion about how population genetics open perspective for a sustainable approach of the seaweed selection and the breeding programs. In Europe, like worldwide, great expectations are made for seaweed aquaculture. And from food safety to the reduction of carbon emissions, artificial selection of the best seaweed appears to be the best strategy. This is supported by many recent works from basic and applied research programs, and examples from Asia during the last decades confirm that the ambitious perspective of developing selection program is a real actuality. The case of the sugar kelp, Saccharina latissima, is particularly interesting because first, it's the most cultivated species in Europe and its life cycle is relatively well controlled. Among the many impacts of Saccharina cultivation identified in the scientific literature, I will only treat here the consequences of the release of reproductive material and the local genetic diversity, and then the stability of the wild population around the farms. And this phenomenon is called the genetic pollution. It is now well known that from a place to another, the size, the chemical composition, and the reproductive period is varying within a same species. And for example, here are shown differences between 3 sugar kelp populations in France, 2 in Brittany and 1 in the north of France. And in North Brittany, the blue and green dots, individuals show longer blades with a wider reproductive period than in the north of France, the orange dot. From a population genetics perspective, each population can be considered as a specific genetic pool. This means that gene transfer between population is rare, and that the genetic characteristics of each population are unique and closely linked to its own environmental conditions. In other words, for this species, the effect of natural selection is strong, and the algae are highly adapted to their local environment. A first question then arises, and one of Peter's talk about earlier, is can a seaweed survive and have the same characteristics if cultivated elsewhere? This strong pattern of local adaptation also asks the question of the consequences in terms of reproductive success for controlled and uncontrolled crosses. You know that the success of crosses is strongly correlated to the genetic distance between parents. And while for some species, males and females that are highly genetically different mate better, for other species, crosses work better when male and female are genetically similar or nearly so. So this pattern is important to know for each species of interest because the perspectives of artificial selection will strongly depend on this compatibility between genetic distant or genetic close related individuals. So to estimate the optimal genetic distance between males and females in Saccharina latissima, controlled crosses can be made in well plates. Each well will contain 5 male and 5 females, for example. As the genetic distance is correlated to the geographic one, these crosses were qualified depending on the origin of isolated males and females. This means that we know if they come from a single algae, the selfing crosses, from different algae of a same population, the within crosses, or from different populations, the between population crosses. Our results show that selfing crosses produce more young algae than within crosses, and that the worst cross mates, male and female, from long distance populations. So according with the hypothesis of a strong local adaptation described earlier, We know now that for Saccharina latissima, an outbreeding depression occurs and then limits the rupture of adaptative genetic complexes. But what are the consequences for the cultivation of Saccharina latissima? The release of reproductive material from farms may have consequences on the local genetic diversity and then the stability of the wild population around the farms. Whatever if genitors come from a local on the left or foreign population on the right, the cultivated seaweeds represent a small genetic diversity compared to the local populations. As a consequence, the release of reproductive propagules or fragments will represent a huge number of new genitors escaped from the farms and with a low genetic diversity that will compete with the wild individuals for reproduction. If each of these two strategies is repeated across generations and considering either any inbreeding or outbreeding depression effects, as explained before, the consequences are at least a more or less fast loss of the local genetic diversity and at worst the total extinction of the local population. We suppose now that Saccharina latissima works like the following scenario. Mainly supplied from local wild populations, the genetic diversity in farms is very low. While no inbreeding depression is expected, the main risk of genetic pollution would be a dramatic reduction of the local genetic diversity by only fixing the cultivated genes. In another hand, the detected outbreeding depression would limit the panel of selection programs targeting the combination of characteristics from distant populations, even if not possible, these crosses are supposed to produce seaweeds with a low reproduction success, which is not suitable with agronomic yields objectives. So, from all this knowledge about Saccharina latissima population genetics and mating patterns, we can propose the following strategy for sustainable sugar kelp aquaculture. First, focus on selection programs on local traits. Second, farms should be more representative of the local genetic diversity. And third, for Saccharina latissima, the local scale should be limited to few kilometers. Some collaborative initiatives for recommendations or guidelines have already pointed all these hypotheses and scenarios at different scales, from national to European or more larger scales. And so the need of a deeper knowledge about the reproduction strategies and the local scale of each species of interest before going too fast. To conclude, the two questions that need to be explored are, first, why is it necessary to think about selection programs at the local scale? Answers would allow us to preserve the identity and the autonomy of each farm, because the history of agriculture taught us that this is the key of resilience and sustainability. And the second question is, what is it necessary to have for sustainable development of selection programs at the local scale? First, a deeper understanding of the genetic diversity and reproductive patterns of each species of interest. A public and private collections of wild and cultivated strains to preserve wild genetic diversity and ensure a supply of high quality and suitable breeding material. And third, we need tools for a common and easily accessible database of genetic resources as the FAO AquaBreeze project. And thank you for your attention.